Good evening, everyone. Thanks for coming on this. Thank you all for coming on this Tuesday evening. We're very, very happy to have Professor Jonathan Karr with us tonight to talk to us on technologies for scalably modeling and simulating whole cells. Professor Karr is a fellow at the Icahn School of Medicine at Mount Sinai in New York, and his lab focuses on pioneering increasingly comprehensive models of bacteria and human cells, as well as developing the computational methods and resources needed for their mission. He did his PhD at Stanford in the lab of Marcus Covert, where he developed the first comprehensive whole cell computational model in a publication described at the time by Cell as one of the most significant papers in their 40-year history. He's the recipient of numerous accolades, and we're really honored to have such a pioneer and expert in the field with us tonight. So thank you once again, Professor, for joining us. Can't wait to hear what you have to say.
All right, well, thank you very much to Krishna and the Society for inviting me. Like Krishna introduced, I'm going to try to share with you how we have been thinking about accelerating the construction and simulation of whole cell models. My personal opinion is that this is a highly multidimensional problem, and so I'm going to try to share with you how we're thinking about several dimensions of this. As a result, I'll go a bit quickly through some of this, but we can always find a time separately if you're interested to chat more about anything that I bring up today. So first of all, I think it's important to understand what's motivating more comprehensive models. So from our point of view, we think that there's two primary things which are driving the need for more comprehensive models of biology going forward. First of all, we think that there's immense need for a more precise, more personalized approach to medicine. And from a decade of sequencing numerous individuals, we now know that one of the biggest opportunities to deliver more precise and personalized care to individuals is to account for all the genetic variation which differs between each individual person. And we know, again, from sequencing that this genetic variation can occur essentially anywhere in the genome. It can affect any gene, any pathway. And so if we want to be able to reason about this in a general way that applies to potentially any person, any disease, we think that this requires much more comprehensive models which allow us to reason about the connection between genotype and phenotype. And very similarly, there's immense desire to be able to harness various properties of biology as machines for various industrial and medical applications, such as detecting disease tissue in situ and maybe synthesizing drugs locally at the site of disease to minimize adverse effects caused by otherwise distributing drugs broadly throughout the body. And so there's, I think, a desire and a need to engineer biology or design biology in a rational way, similar to the way that computer-aided design systems are used to design many other complex systems. But we think that this requires more comprehensive models that could power such computer-aided design systems for biology. So our viewpoint is that there's at least a couple of major applications coming in the future which need a new kind of model, which up to this point has really not existed. So we think that we need to build models that represent entire cells, that represent their entire genome, all of the genes inside the genome, all the other regulatory elements that regulate the expression or function of those genes, all the molecules that those genes code for, and so on. We think that this has to be done over the entire life cycle of an organism. And for several reasons, we think that these models need to be mechanistic. For one, we think that mechanistic models are important for helping us capture the interactions between molecules inside cells. Second, for synthetic biology, we know that we're going to need to be able to make specific recommendations about particular sequences that can be constructed to form the backbone of a synthetic cell. And so this requires at the very least a very kind of fine-grained model, which is much finer grained than typical data-driven models that we see today. And third, we think for the purpose of credibility in precision medicine that it will likely be important to be able to provide a mechanistic interpretation of why a disease is likely to progress in a particular way in an individual, or why an individual should be given a particular course of therapy or combination of therapies. And so we're interested to build models which make dynamical predictions about the physiology of individual cells, as well as can capture the stochastic variation or the stochastic dynamics of the behavior of individual cells. So several years ago, we challenged ourselves to try and build the most comprehensive model of a single organism that's possible. And so we decided to focus on the simplest possible representative example of this class of problem, and that led us to focus on trying to model Mycoplasma genitalium, a small gram-positive bacterium, which in particular has the smallest genome among all known freely living organisms. And so what we were able to show is that through a combination of methods, which I'll describe in a few slides, we could build the first model that's able to capture the characterized function of every annotated gene product. So this animation illustrates the predicted life cycle of one in silico cell. The top left graph illustrates the predicted growth dynamics of the cell. The top right illustrates the size and shape of this cell as it grows. It will elongate, and then it will ultimately pinch to divide itself at its septum. The middle left panel illustrates the predicted metabolism of the cell. The middle right panel illustrates a particular protein complex which forms at the origin of replication to separate the two strands of the chromosome to allow the initiation of DNA replication. The bottom left illustrates the predicted location of every DNA binding protein throughout the life cycle of the cell. And the bottom right panel depicts the predicted abundance of each protein mapped onto its position in the genome. So to sort of summarize, what we've been able to show is that we can construct comprehensive models, at least that leverage the data that we have for very small organisms. And we've been able to show that we can use such models to provide insights into biology. We had a collaboration with Timothy Liu's group at MIT, where we showed how we could use these models to help design cells, and in particular to calculate the metabolic costs of synthetic gene circuits. We've explored how this kind of models could help us repurpose drugs among distantly related organisms. So we've been able to do this, or we've explored a combination of methods to enable the construction of such more comprehensive models. One of the cornerstones that we have based at least our approach to also modeling on is integration of numerous types of data, each of which provides information about a specific aspect of molecular biology. The second major cornerstone that we've based our approach to wholesome modeling on is to combine multiple models of individual aspects of biology or of molecular biology into a single model. There's a variety of reasons that we have enjoyed this approach. One thing is that it allows us to take what would otherwise be a very large model and break this into much more manageable pieces, which we can build, calibrate, and test and debug separately. And so that, in turn, promises to potentially make it easier for individual research groups to build separate submodels individually, and then to combine them into a single model which has contributions from many individual investigators. A third thing that we have made as a cornerstone principle, at least of our approach, is that we should efficiently simulate models of entire cells by co-simulating multiple algorithms which are best suited to the different scales which are relevant to molecular biology. And to give you some rough intuition for how this works, basically what we do is we take a long simulation that we'd like to execute. We break this into small time segments. And then at each time segment, we take a set of variables which represents the instantaneous configuration of the in silico cell that we're going to simulate. So this represents things like the copy number of RNAs, the copy numbers of proteins, the localizations of proteins, the locations of DNA binding proteins on the chromosome, the sequences of transcripts that are undergoing transcription, and so on. We take these states and essentially pass them to all of the submodels, and then we execute each of these submodels using different algorithms which are appropriate to the scales relevant to each submodel. So the key thing is that we can then simulate submodels using different simulation algorithms that are relevant to the different scales. So for example, we can model transcription, the stochastic dynamics of transcription and translation, where the noise in the counts of individual RNA or proteins are relevant. At the same time, we can model metabolism much more coarse, or simulate metabolism much more coarsely using a method like flux balance analysis, which is appropriate to the concentrations of metabolites and the fluxes of reactions, which are at much higher concentrations and much higher fluxes than the counts of RNAs and the rates of transcription. And so essentially what the execution of these algorithms does is it computes something like a change to the state of the cell, and then we sum up these changes across the execution of all the submodels to compose the next state of the in silico cell. And then what we do is we just repeat this process many times to build up the predicted dynamics of an entire simulation of an in silico cell. So to summarize, the way that we have been able to pilot wholesome models is through a combination of data integration, model composition, and co-simulation of multiple algorithms capturing different scales relevant to molecular biology. Now, despite that progress, however, there's still a substantial amount of molecular biology and physiology or phenotypes that even our models have not been able to capture. So this graph illustrates a particular set of phenotypes that our model has not been able to capture. So the red illustrates model predictions, the blue illustrates experimental measurements, and this highlights the discrepancy between the two. So each vertical column here represents the predicted and measured phenotype of a single gene deletion strain of Mycoplasma genitalium. And where you see discrepancies, so where the blue and the red don't overlap, these are places where the model is not able to accurately capture the phenotype of a single gene disruption strain. So what we take away from this is that although we've built maybe more comprehensive models than what has been published in the literature, we still have a long way to go to be able to predict all of the phenotypes that might be relevant to engineering mycoplasmas or might be relevant to their pathophysiology. And of course, these are very small cells. We still need to figure out how do we scale this up to much more complex cells, ultimately up to human cells, to be able to deliver precise medical care based on models of human cells. And so what we've been doing over the last several years is thinking back on this workflow that we piloted to build a model of Mycoplasma pneumonia, or Mycoplasma genitalium, and think about how do we scale up every aspect of this to support much more comprehensive models. And so we've been thinking a lot about what are the bottlenecks to this workflow, and how do we devise computational methods and software tools to get around all of these bottlenecks. So, so we felt that it's important to really think concretely about exactly what are the challenges that we have to solve in order to be able to do this. So one of the first, in no particular order, one challenge that we need to address is that within a single model, we have to be able to capture a diverse range of molecular biology with a diverse range of network architectures and diverse range of scales that are involved. So we need to be able to capture networks that might have a hierarchical structure, like a transcriptional regulatory network might. And at the same time, we might need to capture networks that have more of a small world network architectures that might look like what that might capture the structure of a metabolic network. Another challenge is that we believe that we will likely need to capture the combinatorial complexity which is inherent to biochemistry. So a couple of places in particular where there's intense complexity is in the generation of multiple isoforms of each gene resulting from alternative splicing, and also in the generation of many proteoforms resulting from a single protein from differential post-translational modification. And similarly, there's an equal complexity which is generated through differential post-transcriptional modification of RNAs. So together, these processes basically create a huge range of molecules which have, to some degree, differing behavior and differing abilities to catalyze reactions. And so this range of chemical diversity is real, and it can affect phenotype, and therefore there's at least, I think, some need to capture this combinatorial complexity. But it introduces a lot of challenges in modeling. A third challenge that I've already mentioned is that we need to, within a single model, capture a diverse range of scales. The reason that this is challenging is that it means that we can't simply use the conceptually simplest stochastic simulation algorithms to simulate an entire cell. I mean, in principle, you could. The problem is the computational cost is too high. And so instead, we have to resort to hybrid methods. But hybrid methods is still a relatively undeveloped area of mathematics. More practically, challenges include the fact that there's no single experimental modality that we can use to calibrate an entire model. Instead, we have to try to use multiple different types of experiments and somehow stitch them together. And worse, the different data that we might want to use is scattered throughout the internet. It's scattered throughout different journal articles. It's collected by different fields. So there's not a lot of standardization that makes it easy to combine data sets together. And with a few exceptions, the data is generally incomplete. So in a typical metabolomics experiment, you might expect to be able to measure at most maybe 10% of a bacterial cell in a single experiment. And even that, there's probably only a handful of examples in the literature of data sets that are even that large. So we've been thinking about, in the context of these challenges, how do we scale up this workflow to support more comprehensive models of the biochemistry of individual cells. So in the next 25 minutes, what I'd like to do is tell you about some of the technologies that we've been developing to alleviate the bottlenecks to building more comprehensive models of cells. So several years ago, when we were starting to think about how to approach this problem more systematically and scale up to larger models, we sent around a survey to the biomodeling community, and we asked the community, what do you think are the major bottlenecks to progress? And the most frequently cited problem was lack of access to the data that's necessary to model biology. And this was true not only at the molecular biology scale and relevant to cell modeling, but other investigators working at other scales reported very similar issues. So as an example, let's say we wanted to model the dynamics of a metabolic reaction. We would likely need, at the very least, information about concentrations of metabolites. It would likely be useful to have data about abundances of enzymes. It might be useful to have some information about the subunit composition of enzymes, information about reaction kinetics. And a lot of this information, there is a lot of information about all of this, but it's scattered in many different places. And of course, as we've already discussed, to model an entire cell, we need vastly more information. Beyond this, we would like information about abundances of RNAs. We would like to have information about how genes are regulated, and so on. And as we think about even more diverse data, then the problem of where to find it becomes even worse and worse, because it becomes even more and more scattered throughout the internet and the literature. So because this is a huge bottleneck to not only our efforts to model biochemistry, but others as well, we decided to approach this by developing a toolkit that we call Datenator, which is available at datenator.info, which tries to make it easier for investigators to find the data that they need for these kinds of meta-analysis of biochemical information. So the first kind of part of Datenator is an integrated database of various types of molecular data, which have been integrated from a variety of different sources. This includes currently concentrations of metabolites, information about modifications, half-lives and localizations of RNA, information about abundances, modifications, and localizations and half-lives of proteins, and rate constants of metabolic reactions. Now, essentially what we've done is basically take a variety of different types of data. We have obtained them from a variety of different sources. We've then parsed them all, normalized them, put them into a consistent set of units and identifiers, provided consistent metadata for this, and essentially assembled a huge cloud of data, which is kind of uniformly represented and uniformly annotated. And this cloud of data encompasses data from a huge number of molecules, a large number of reactions, and many different organisms. And so the next thing that we need to do is to make it easy to search through this data to find data that's relevant to any specific project where we would like to understand a particular set of molecules or reactions in a particular organism, and maybe even in some range of experimental conditions. And so what we've done is essentially built a search engine that allows us to search through this large and high dimensional, but also sparse data space. So like other search engines, or like other databases, the first thing that users do is they type in a description of the molecule or the reaction that they're interested in, and the organism that they would like information about that molecule or reaction. And users can search by names of molecules or identifiers, or they can use, search by smile strings, inking strings, and reactions can be searched similarly. And then Datenator displays a set of a list of molecules and a list of reactions for which we have data, sort of disambiguating the user's query. And next, users can click on any of these results, at which point they're presented with tables of potentially relevant information to the molecule or reaction and the organism that they're interested in. And so essentially what we do is we take this huge cloud of data and we try to kind of filter that down to data that's potentially relevant to the molecule or reaction that the user is looking information for. But because the data set is sparse, so we don't have information about literally every molecule, or literally every gene in every organism. But we often have data about maybe structurally similar metabolites and perhaps closely related organisms. What we do is we bring down data from not only the molecular reaction that the user was interested in, but also data from structurally similar molecules or mechanistically similar reactions. And we pull down data not just from the organism that the user was interested in, but also data from taxonomically similar organisms. And so basically this provides a cloud which is still somewhat large. And so next we provide filters to allow the user to use other contextual information they have about their research project, which is perhaps difficult to encode in a web form, and provide investigators sliders and other controls to be able to narrow down these clouds of data down to data that they think is most relevant to their investigation. So for molecules, for metabolites, investigators can narrow down these sort of data clouds by their 10-amino similarity to the measured metabolite. Measurements can be narrowed on the basis of the organism that was characterized using the NCBI taxonomic tree. So we can filter down to measurements that are only from the same species, or from maybe the same genus, or from the same class, or all the way up to members of the same kingdom, or even just any measurement of a similar metabolite or gene or protein across any organism. And we have similar filters for characteristics of environmental conditions as well. So basically what this allows you to do is, without having to directly go to hundreds of articles and maybe tens or hundreds of databases and try to pull out data that's going to be relevant for a particular project, this provides one place where you can systematically search for data that's relevant to modeling. So where we have begun to use this is to pull out all the information that we've aggregated about the metabolite concentration, the concentrations of metabolites in E. coli, which provides us with information about concentrations of over a couple of hundred metabolites, merges all this information together, and now we're using this to construct a higher resolution model of the biomass of E. coli as a component of a flux balance analysis model of E. coli. So essentially, what this has allowed us to do is better constrain the flux predictions of a flux balance analysis model of E. coli. It also highlights gaps in the model. So there are metabolites which existing models cannot explain the production of, and this has led us to then add additional metabolites and reactions to what is thought to be a very comprehensive model of E. coli, but has actually overlooked some areas of metabolism by neglecting a particular dimension of metabolism. So one of the long-term challenges to integrating data for models is that data is often published in a very heterogeneous way. So at least in our field, much of the data that's relevant to understanding biochemistry is published as supplementary spreadsheets or Excel documents attached to journal articles. And these documents are to some extent structured, but to a large extent not really structured in a way that's conducive to modeling. So they can, for example, have multiple dimensions effectively encoded into them through the use of multiple layers of headings. They can involve many different data types. There can often be examples where there are multiple tables that are linked together without any obvious connection or software to help make these connections. Due to the lack of software tools for working with such data, there's often bugs in these data sets, which can make it difficult to interpret them. And so in contrast, as a modeler, as someone who wants to do meta-analysis and combine multiple data sets together to overcome the inability to characterize a cell with one experimental modality, what we're looking for is structured data. We want explicit, we want to really understand the connections between different parts of the data set. And we want high quality data, and we want to understand what the data types are and the semantic meaning and so on. And so what we have built to facilitate this is a set of tools that we call object tables, which allows us to basically link in a fairly easy way spreadsheets which have a little bit of additional annotation to a set of object-oriented data structures that make it easy for us to do computations on experimental data, particularly to merge data from different sources and different types together for meta-analysis. So this is available at objecttables.org. This, because it's motivated by and developed by investigators who work in the area of molecular biology, in particular, it provides a lot of support for types of data that are relevant to molecular biology and biochemistry. So this is available in several different user interfaces. There's a web application, there's a command line program, there's a Python library. The Python library is the most flexible of these, and this is the primary tool for merging data sets together. But even the simpler user interfaces provide tools to take multiple data sets, to do things like validate them, to find errors, to compare them, merge them, visualize their structure, and more. So as an example, we have used object tables to take two different data sets which provided thermodynamic and kinetic information about central metabolism in E. coli, and use object tables to parse them individually, merge them into a single integrated data set, and then layer that combined data set onto flux balance analysis flux balance analysis model of the metabolism of E. coli to further constrain flux predictions made by the model. So the graph on the right illustrates reactions. In purple illustrates reactions which we could constrain both with kinetic and thermodynamic data. So this is where there's intersection between data sets which were published individually by different research groups. Green and blue illustrates places where we're able to constrain a model based on data from just one of the two data sets individually. So we think that object tables is potentially a key ingredient to enabling not only data creators to provide data in a format that can be more easily used by computational scientists or other investigators for meta-analysis, but we think it's also a tool for modelers to be able to take experimental data and easily reuse it and combine it. So another issue that comes up in wholesome modeling is that we would like to be able to work with we would like teams of investigators to work together to be able to construct wholesome models. And this can be one thing that's challenging about doing this, especially with large models that represent large numbers of metabolites and reactions, is that it can be difficult to understand exactly what a model developed by someone else means. So the typical way that this is approached in our field is that proteins, for example, would be annotated using uniprot terms. And where this becomes problematic is, let's say that you would like to model a signaling pathway and there are multiple phosphorylated forms, or even maybe species that represent double or triple or phosphorylation of the same protein. All of these forms of the same proteoforms of the same protein would typically be annotated using the same uniprot identifier. And so this makes it difficult for different investigators to come along and understand what a model means, much less compose it with another model. So we've developed a couple of tools called BP forms and BC forms to allow us to concretely describe the physiological molecules that are involved in molecular biology and which need to be captured by computational models. Essentially, what BP forms does is generalize the FASTA format, which is commonly used to describe how DNA, unmodified DNA, RNA, and proteins are composed of the canonical 4 nucleotides for DNA, the canonical 4 nucleotides for RNA, and the canonical 20 amino acids for proteins, and generalize this to describe how physiological DNA, RNA, and proteins are composed not only just of those canonical nucleic and amino acids, but thousands of other modified nucleic and amino acids, cross-links, and other biochemical features. So this allows us to, in a fairly compact way, as illustrated at the bottom of the screen, be able to describe polymers, and there's a related grammar for describing complex, and have a precise chemical meaning illustrated by what you see at the graph at the top. So to make this easy for investigators to work with, we developed some software on top of this, which allows investigators to parse the descriptions of models that are encoded in this sequence, to calculate properties of molecules that are described using BP forms and BC forms, such as their molecular structure, their chemical formula, molecular weight, export them to other formats, generate visualizations, and so on. So how can you use this for modeling? So as an example, this is a model of the MAPK signaling pathway, which was published by Boris Kolodenko almost 20 years ago. And the model represents several phosphorylated forms of proteins and some doubly phosphorylated forms. So what we can do is we can go through this model and use BP forms and BC forms to systematically annotate the semantic meaning of all the molecules represented by the model. If we do this, we can then use the software to calculate the chemical formula of all the molecules, and then use that to mass balance all of the reactions. And so this then kind of immediately identifies the fact that none of the reactions are mass balanced, and highlights all of the metabolites which have been missing from the model, which could provide connections, say, to a model of metabolism that could be coupled with a model of this signaling pathway. Another thing that pops out by annotating the model this way is that there are other singly phosphorylated forms of these proteins which are also missing from the model. And so BP forms and BC forms allow us to systematically identify missing forms of proteins which may be relevant to the physiology and potentially need to be included in a model. Another thing that we can do with BP forms and BC forms is if we annotate this model and we annotate other models, then we can use this to systematically identify overlaps with other models. So if we, say, took a database of models and we annotated all the models in the database, we could then systematically look for overlaps between models and use this as a starting point for merging models together. And of course, we can combine all this together. So we can mass balance, identify missing metabolites through mass balancing, identify missing macromolecules through looking at potentially missing combinations of modifications, and systematically map models onto other models as a kind of substrate for model composition. So one of the other challenges that I introduced, or one of the other principles that I introduced for our approach to whole cell modeling is to build models by composing models of multiple submodels. And then I introduced the idea that each of these submodels could potentially be executed or simulated at the scale relevant to their biology. So this requires composing a model together and then code simulating the relevant algorithms that are desired for each individual submodel. So we have shown how this can work in principle, but to scale this up, there really needs to be methods which formalize these ideas and software tools that implement them. So over the last couple of years, we've developed a language called WC-Lang, which provides a relatively simple way of describing such composite multi-algorithmic models in a fairly direct and simple way. And one of the other key features of WC-Lang that we're still developing is the following. So as I mentioned, we think that whole cell models have to represent an enormous range or enormous number of molecules involved in biochemistry. So one example is, let's say we wanted to model the transcription of each individual nucleotide in every RNA in a cell. So we could, and the kind of canonical way that this has been approached in the past would be to enumerate every possible elementary produced. But of course, this has lots of problems. This would involve an enormous number of species and reactions. And even that wouldn't be able to account for issues like errors in transcription or skipping, which would necessitate an even larger state space. And so even if you could enumerate this, it would be difficult to maintain. No other investigator would feasibly be able to understand this. And instead, what we want is to be able to describe molecular biology in a way that's much closer to the way that we would maybe think about it or describe it in an article or in a textbook. So what we'd like to be able to do is describe molecular biology more like as a set of patterns which can generate the individual species and individual reactions outlined on the previous slide. And so the other major feature that we're still finishing as part of WC-Lang is the ability to describe models using these kinds of high-level patterns. You can basically think of as like an abstraction over this reaction network formalism of describing biology. So this is what reaction patterns would look like for descriptions of transcription on the first line, the turnover RNA in the second line, translation of proteins in the third line, turnover of proteins on the last line. Now, to simulate these models, we've developed a tool called WC-Sim, which allows us to combine together in a single simulation the execution of multiple simulation algorithms together. And it's architected in a way which can accommodate essentially arbitrarily many time-driven simulation methods, and this is in contrast to previous approaches which have nearly all focused on combining just two types of simulation or co-simulating just two types of simulation algorithms together. But for wholesome models, we felt that there aren't just two scales involved, there's really multiple scales. And so there's a need to combine not just two simulation algorithms, but potentially multiple simulation algorithms targeted to the various scales. And so WCSim is one of the first tools that allows this to be done in a systematic way. So as I mentioned, we think that collaboration will likely be an essential part of building more comprehensive models of biology. The reason that we think this is that we believe that such models need to be able to harness the expertise about different areas of molecular biology that are possessed by different investigators around the world. Unfortunately, I think despite various efforts to standardize modeling, it's remained difficult for modelers to collaborate on these kinds of mechanistic models. And so to alleviate, in particular, I think it's remained difficult not so much to collaborate on models, but to collaborate on simulations of models. And so what we have done over the past year is developed several missing standards, or to fill in gaps in the existing standards for simulation tools, to provide a more comprehensive set of standards for describing simulation experiments. And then we've developed a registry of simulation tools that we call biosimulators, which is available at biosimulators.org. Essentially what this provides is a place where investigators can go and find simulation tools that are capable of executing a specific model. And then many of these simulation tools are available as standardized Docker containers. That includes several simulation tools for continuous and discrete kinetic modeling, constraint-based modeling, logical modeling, rule-based simulation. And the key thing is that all of these simulation tools, due to the new standards that we have created and the gaps of standards that we've been filling, all of these tools are available through the same interface. So this greatly simplifies things for investigators. So if you receive a model from a collaborator, you don't really need to learn a new tool. You can just use the same interface that your favorite tool provides to execute another type of model that you might not be so familiar with. And because these simulation tools all have the same interface, this has allowed us to provide a web application where we can provide a single web form where an investigator can come, choose a particular simulation experiment, select a particular simulation tool, irrespective of what modeling formalism or model format or simulation algorithm may be required to execute this, and then click a button and have a web service execute that simulation for you and display the results of that in a common web application. One additional piece that we're building on top of this is a repository for sharing simulation studies. This will be available at biosimulations.org. So where is all this going? So in our group, we have been taking these tools which try to address individual bottlenecks to the construction and simulation of wholesome models and combine them to build both more comprehensive models of similar bacteria, as well as to pioneer more comprehensive models of human cells. So on the human cell front, we've been able to take the consensus metabolic reconstruction of human metabolism, and then layer on top of this several different types of data to build a quantitative dynamical genome scale model of the metabolism of human embryonic stem cells. And now we're working on combining that with several stochastic submodels of the stochastic dynamics of the synthesis and turnover of RNA and proteins. So hopefully later this week, if not next week, we'll have a preprint up on a model which represents the same biology that I described, but for Mycoplasma pneumonia, which is closely related to Mycoplasma genitalium, but a bit better characterized. So in summary, we think that there's a key need for whole cell modeling going forward, both for precision medicine and for synthetic biology. We think that whole cell modeling is becoming feasible through a combination of methods that center around the integration of data, the composition of models, the co-simulation of simulation algorithms. And we think we've developed a key set of tools, including Datonator, WC-Lang, and WC-Sim, which address some of the key bottlenecks to applying these composition ideas and using them together for more comprehensive models of cells. So with that, I just want to thank all the members of my group and our collaborators and our funding, which has contributed to all of this work. And thank you very much for your time and attention.
Yeah, I mean, thank you so much, Professor. It's a really, really interesting talk to hear about the problems that need to be addressed and the tools you developed to solve them. So I think we'll open the floor to any questions now. I think there's already one in the chat, so either feel free to put your question in the chat, or you can turn your camera and microphone on, and you can feel free to ask it face to face. But if we start with Chris Davis's question, who asks, I may have missed this, but can you use this model to determine the impact of a drug on the cell? If so, how do you go about to identify which biochemical pathways are added, interrupted, or modified over the simulation cycle?
Yeah, I mean, in our own work, we have not focused a lot of attention on drugs, but there are other people who have, such as Mark Burke-Wissel, and at least in the United States, there's a project called Links, which has attempted to catalog the molecular effect of various drugs. So what they do is they basically have like a panel of drugs, and then they conduct the same molecular and physiological measurements to assess the impact of those drugs on a few cell lines. And then all that data is, in theory, available for modeling. And Mark Burke-Wissel and Peter Sorger in particular have been involved in trying to model the data out of the Links project. But basically what people try to do is distill out the molecular mechanism of those drugs and encode that into a reaction network formalism, as I outlined. And then the impact of the drugs can be kind of treated just like any other molecule from a mathematical or computational perspective.
Thanks for that. I should also mention there is a raised hand function here in the bottom right. So I think Bobby, you raise your hand. I feel free to have a go.
Yeah, sorry. Yeah, sorry for the names. Got a screen name going on there. Yeah, thank you very much for your talk. It's been really interesting, thought-provoking. My question is on, and I'm sorry if I've missed this during your talk, but on spatial dynamics. I was wondering, does the modeling framework that you've described here take into account kind of the spatial distribution of molecules and interactions? And if not, if so, how does it do that? If you can give a quick explanation, and if not, would it be possible to include that if you're going to be scaling up this model to take into account, eukaryotic cells where this spatial dynamics would be really important?
Yeah. So in at least in our work, we have and various other people have done very similar things. We have felt it's essential to try to capture the subcellular localization. But in our work, we've done this in a fairly coarse way, primarily because we don't want we don't want computations on space to just overwhelm, to drive up the computational cost. And so in like a model of the human cell, we would want to see compartments that, and what we have are like compartments that represent the nucleus, the membrane, the cytosol, the endoplasmic reticula, the mitochondria. So that's like the level of resolution that we've been working with, like the canonical named organelles. But again, to kind of point out some of the work of other people, one group in particular that has been focusing a lot on the role of the spatial organization of cells and the diffusion of molecules inside a cell and how that impacts physiology is Zan Schulten at UIUC. And but the like flip side of that approach, as I alluded to, is that the computational cost is quite a bit higher. So the let's kind of give you some rough intuition. I mean, in theory, you could maybe try to simulate a cell using molecular dynamics and represent every atom. The problem would be that you would only be able to simulate a very short length of time, and that might be so short that it's not particularly relevant to cell physiology. So they operate in some in sort of a middle ground where they're running simulations that are very similar to molecular dynamics, but done in a coarse-grained way where the particles represent either entire proteins or subunits of proteins. But still, the computational cost is high, and so the length of simulations is low. So that's just, like, I think at the moment an inherent trade-off due to the primitive nature of the methods. So I think what myself and probably others would like to see is a future in which these more genomic-focused perspectives on wholesome modeling that my group and a couple others have been pursuing is merged with this more physical, spatial aware vision of wholesome modeling that Zan Schulten and a couple others are pioneering.
Right, thank you very much.
Great, so I think Mark, next, if you would like to ask your question.
Yes, thank you very much. Thank you, Professor Karr. It was very interesting. It's a very fascinating topic. And I'm actually doing a PhD in protein solubility. And when I read about your work, I was immediately thinking about, okay, how close or maybe how far is the model to be capable of seeing what slight changes in bio or physicochemical properties of proteins can have on the overall system. So I'm thinking a little bit about proteomics. If, for example, one protein is overexpressed or there's a mutation that a post-translation modification is changed, something like this, how this downstream affects the overall cell.
So we're very interested in this particular question, and this is in many ways, particularly you mentioned post-translational modifications. And I think post-transcriptional modification is very similar. This is really what's motivating this kind of rule-based thinking and this idea of complexity. So we know, I guess my perspective is we know that there's huge diversity in the protein population that exists in a real cell. And we know that we don't have good machinery to keep track of this. So we're trying to develop the computational methods that would allow us to do this. The hole in the methodology, I would say, at the moment is that we're, I think, pretty close to being able to track the individual species, to be able to say, like, this protein, this variable in a model represents a protein that has this particular set of post-translational modifications. And we're, with BP forms, we could say, okay, this is, we could, in a text format, like describe what that is, and then we can convert that into, say, smiles or inky, and give you a very precise chemical definition of what this means. Where there's a big hole here, though, is, and we don't really have, I think, a great idea about what to do about this, is, what's motivating this is that these molecules all behave differently. They, for example, might catalyze reactions at different rates. That's why we want to capture them. And there's plenty of examples where we know, like in the case of transcriptionally modified RNA, we know that like various modifications have particular purposes. But at the same time, we don't have good quantitative data to say, okay, if we see this modification, then that means that the enzyme has this turnover rate. So we, I think, somehow need to figure out still, how do we connect what the structure is to a parameter that would affect the catalysis, or the reaction catalysis that reaction, or that enzyme serves in the model. I mean, maybe a role here, a solution here would be to couple in some approach from structural biology, but I'm not sure what that would be. I think this is a big open area. I mean, what I'm seeing in the literature is even this concept of a proteoform is relatively new. The term proteoform has only been around for a few years. The structural methods to catalog it are just emerging. Things like BP forms to be able to describe it are just emerging. And so the functional characterization of all of this, much less the modeling, is still, I think, coming along.
Okay, thank you very much.
Okay, Manas Kohli. Sorry if I mispronounced your name.
That's all right. Thank you for the talk. It was really interesting. I guess I just had a question with regards to sort of, well, I had like a two-part question. One is that how exactly is your, like, I guess how robust is your model when it comes to like analyzing mutations? And in particular, like if you can capture sort of redundancy in biological networks. So like, I mean, one example I'm just thinking of is like, if you have like, a knockout of a particular gene, and now metabolic basically kind of pathways are reprogrammed, I guess, at different scales or things like that. So like how can your model kind of capture that, or are there any kind of things you need to do further before you can capture that kind of complexity or robustness in your whole cell models?
Yeah, and in the approach that we're taking, you can certainly represent redundant pathways. So there's nothing which says that it's necessary to only have, say, one enzyme that catalyzes each reaction. So you can capture things like knockout of genes and the fact that the cell still lives. Or you can capture essential genes where, which are truly essential, where there is only one protein which is able to code for a specific function. I mean, the mathematical formalism allows you to capture either one. I mean, mycoplasmas, that said, are like kind of a unique area of physiology where, because of their reduced genomes, they don't have a lot of redundancy. But that's a real aspect of their biology, and so that's intentionally captured by models of mycoplasmas. But in human metabolism, for example, that's not the case. There's a lot of redundancy. So, our perspective would be like, whatever the biology is of the organism we're interested in, we're going to try to mimic that in the model. And the mathematical foundation of this can accommodate either way.
Okay, cool. Thank you.
Dennis. You there, Dennis? Well, perhaps I can read out his question from the chat. But he was basically asking about Lewis Wolpert's pioneering work on mechanics of the earliest stages of cell division back in the 1960s. And apparently he died sadly a few weeks ago from COVID. And he predicted in 2004 that 100% accurate computer simulations of whole cells would be possible in 20 years. So then his question is, 3 years before the deadline, was he right? And if so, would it be possible to do so in real time?
I mean, I think we're obviously more than 3 years away. I mean, at this point, yeah, I think we're very far away. I would even kind of hesitate to speculate on how far. I would think we're at least several decades. I mean, at this point, there's only a few investigators even doing this. So I don't think progress is actually even advancing all that quickly. And I guess one thing I do look to is it seems to me that writing a genome is substantially easier than modeling a genome, which is kind of roughly what we're trying to do here. Even writing a human-sized genome is really not possible at this point, and probably at least a decade away. I would think that this is at least several decades behind that, so that would, in my estimation, be at least, say, 3 decades. But of course, like, it's hugely dependent on investment and how many other people contribute to the field. So I think if governments were, and maybe industry eventually was to invest in this in a big way, then that could, of course, substantially change the landscape and greatly accelerate this. But if that weren't to happen, and this continues to proceed with just a few investigators, then this could take quite a long time.
Then does that answer your question about, is that because of? I guess it's
oh, I see. Well, I would say, so I think well, I would say both, but I think the lack of computing power, I don't think that that's like the barrier which is really actively preventing progress at the moment. I think the bigger issue, which is more actively limiting progress, is lack of sufficient data, and even more specifically, just lack of ability to even use the data that already does exist. And we've tried to pull together several types of data with Datator, but there's a lot of other data beyond that, and it's very disorganized, so it's difficult to make use of all of that. And the second thing I would point to would be lack of methodology and lack of easily usable software tools. So a lot of the things that, if you look back at like the way that we were doing things in 2012 with the model that we published in Mycoplasmas genitalium, we pioneered some ideas, but it's not packaged up in a way that other investigators could easily use them. And so you need a lot of detailed technical knowledge. And we've been trying to make things easier, but it still requires a bit of technical knowledge because these tools are still fairly primitive. I think to get more people involved, to accelerate this, the tools have to be easier such that you don't need maybe several years of like programming experience to be able to navigate this field. And we're, in my opinion, not there yet. So at the moment, I think it's because of the primitive nature, it is a fairly technical field, and so it's hard to navigate without a particular set of skills for dealing with that primitive nature.
Cool, great. Thank you. All right, I think Bram, you have a question. Would you like to?
Hi, Professor Karr. Thank you so much for your talk about your exciting work on computationally simulating the whole cell. I think it's really interesting, but I was just wondering, if you're trying to simulate a whole cell, surely you would need a lot of data in terms of the proteomics, transcriptomics, probably even the metabolomics. How are all these data obtained? I mean, in recent years, probably these omic tools have improved drastically. But just wondering how you managed to obtain all this data to simulate a cell accurately.
So, well, so originally we essentially just queried the literature rather ad hoc. So we would say, okay, we would like information about metabolite concentrations, and so we would go to Google Scholar or Google or PubMed and just search for proteome-wide data like Mycoplasma genitalium. But it's really hard to, I think, search the literature that way, because we know that the literature is sparse. So we know that we're not going to be able to find every type of data that we might want for, especially for an organism that's not deeply characterized like Mycoplasma genitalium. So you want to be able to somehow search not just for data about Mycoplasma genitalium, but other similar organisms. And it's not so easy to do that using Google Scholar or PubMed. Really, the only way to do it is just to enumerate like every other organism which is similar and stitch them all together with OR operators, or maybe write a program to do this. But then you're just going to have a lot of papers which you somehow have to manually comb through. So more recently, what we've tried to do is be a bit more systematic about this and divide up the problem. So first, we just integrate data. We don't really worry too much about what organism is involved or whether we care about that for a specific project that we have in mind. We just integrate the data that we think is necessary, and then we separately devise tools for searching over this. And by doing it this way, we can't use Google Scholar or PubMed to search in the way we want, but if we put it in our own database and we structure the metadata appropriately, we can then search in the way we want and find data that we need. But this all rests basically on a lot of manual curation of data at this point. So we've been thinking about, and this is kind of where object tables comes in, is how do we standardize how data is produced? How can we maybe get other people involved as a community to curate data? But that's, I think, making progress has been a little bit hard because investigators really love their ability to just publish data in whatever format they feel like. And you know, reviewers of journal articles there's not much incentive really, I would say, for your typical investigator, unless reviewers or journal art editors or some governmental body forces them to produce data in some usable format. They're just going to keep sharing data in whatever format they feel like it, or maybe not even really sharing the data at all.
Great, thank you. And Mark, you have a question.
Yes, thank you. I hope it's fine that I ask another one. It's a rather personal one. So I'm at nearing the end of my PhD, and I'm thinking about what to do next. And the whole cell modeling was always very fascinating to me. And I'm just wondering, because it's such a broad and diverse topic with so many different layers, where so many different expertises are needed, what do you think are the skill set or the basic knowledge that someone would need to join that field of whole cell modeling?
I think in a lot of ways, just ability to write code is maybe one of the most essential skills to have, and to be able to do that relatively efficiently in a disciplined way. So some basic knowledge of like unit testing, continuous integration, repositories. My opinion is if you know those things well, then that allows you to move quickly and experiment and not get tricked up by all the technical issues around using these kind of primitive academic software tools that don't have a lot of documentation, that you would need to do all of this. At least in my experience, this is probably the biggest thing that I've seen people get tripped up on. They don't know this, or they are not really willing to learn it. They have generally not made it very far. They just get stuck on kind of what are, in some ways fairly trivial things. And I think one of the other benefits to that is these are the skills that I just mentioned. These are very, in my opinion, very transferable skills, which are also in strong desire in industry. So I think that there's a lot of value in developing this. And then you can take this and apply it to lots of problems in science. These are not skills that are unique to wholesale modeling. So it sets you up I think, to do a lot of things.
Okay, perfect. Thank you very much.
Okay, if no one else has any more questions, I'd just like to actually, we had a quick dig through the archives, and yes, Professor Liz Wolpert gave a talk in February 2007 on science and religion, actually, while he was a professor of anatomy, I think, at UCL. So yes, sad to hear of his passing. But anyway, it was great to have you, Professor Karr. Thank you so much for giving up an hour and a bit of your time to talk to us. It's really, really insightful and really interesting. And I think actually someone did ask whether the slides that you use would be available anywhere.
I can send them to Krishna. That would probably make the most sense.
Yeah, that would work greatly. But this talk will also be up on our YouTube channel in a couple of days, so the slides will be available there too, I guess. But yes, again, thank you, Professor Karr, very much for joining us. Thank you all of you for spending the evening with us, and hope you enjoyed it. And we'll see you next week, I believe. Thank you very much again, and hope you have a good evening and the rest of the day for you.
Well, thanks for the invitation.